Good evening, everyone. A lot of you know me, those of you who don't, I'm Mike Sullivan. Until May, I was the External Affairs Manager here at Skidaway Institute, and I was called in to pinch hit for Clark Alexander this evening, who is recovering from a bad back of some kind. I'm glad you all, pardon me, found our new venue. I thought I'd take just a moment, because most of you have probably never been in here. As you notice, it's somewhat of an unusual structure. It was built right after World War II by the Robert and Dorothy Roebling, who operated a cattle plantation here from the mid-1930s until the late 1960s, when they donated their land to the state to form Skidaway Institute of Oceanography. They built this barn as a show barn. Roebling was an engineer, as all his family really was. His great-grandfather, John Augustus Roebling, was the designer and builder of the Brooklyn Bridge, as well as bridges all over the, pardon me, all over the country. And that's where, but I'm not going to go through the whole story of Skidaway Institute. Anyway, it was a cattle plantation, cattle farm, and this was their show barn. You see a mosaic here in the middle of the floor. That was just for decorative purposes. They called it Modena Plantation. The door directly behind was where they would bring the cattle in. Around the outside were the cattle stalls, and they faced outwards, and the infrastructure of the facility was here inside, in the interior. And when it was water, there was a mezzanine level, which all the way around. As a matter of fact, until we went to remodel it, there were still bleachers up here for their annual cattle show, which they did every year. They would bring the cattle in, and they would parade them around, and people would bid on it, and people would come from as far away as Europe to bid on their Black Angus cattle. It was basically a warehouse, for lack of a better term, from when we took over in 1968-69 until 2018, when we got money from the legislature to remodel it and turn it into functional space. It was concrete floor, open, pigeons flying around in the rafters, unair conditioned, unheated. It was a great place to prep equipment. You got a big piece of equipment you're going to take to sea, like the lander, you're going to drop over the side of the ship. It's a great place to do it because it was big, it was open, and you didn't have to worry about scratching the furniture. So we lost that. It was also storage. That caused some pain for our faculty when they had to clean out their stalls before the remodeling effort.
And we had all the freezer space in here. That wasn't the freezers for storing samples. That was all in here too.
Yeah. And so we remodeled this and dedicated it in 2019, just before COVID, and turned it into, the idea was to revitalize an historic old building. We have two very high-tech classrooms that allow our professors to teach classes here, as well as have to students here, as well as having students in Athens. In addition, you can also have a professor in Athens teaching a class with students there, and the students here with very easy, back and forth. I say that easy because I'm not doing it. I've heard from these guys that it's not necessarily always a piece of cake trying to when your students are spread over to different locations. But that's what this is. It's the old Roebling Cattle Barn, now called the Ocean Science Instructional Center. Tonight, I'd like to introduce Dr. Marc Frischer. Some of you have heard Dr. Frischer before, talking about black gill, which is something he's been working on. He's expanded that a little bit to talk about pandemics in general, especially in the marine environment. And Dr. Frischer received his bachelor's degree from Washington University and his PhD in microbiology from the University of South Florida. So without further ado, Dr. Frischer.
Thank you, Mike.
Thank you all for
coming and finding us here in this really interesting structure. Our role, some of you may know or not, we used to be, until 2013, we were an independent unit of the university system. And in 2013 is when we joined the University of Georgia, which really changed our culture and put a lot more teaching in. So it was very important that we have a good teaching space. And so that's what this has become. And it's very useful for that. All right, so before I go, I just wanted to just give you a little background of my academic and research interest and background, because I think it'll help sort of understand how I got into this sort of crazy field that we're going to talk about. And if you take home one thing when we start talking about all these terrible diseases that are happening, and coral reefs dying, and seals and sea lions and otters perishing, when we start talking about that, I want you to keep in mind that for the most part, microbes on our planet, microbes in our body, even the disease-causing ones, are really part of a healthy ecosystem. And microbes get a bad rap because we always think about disease. So I just want to put that out right in the beginning. So as we go through this, keep that in mind. So I'm a microbial ecologist. And so what I am interested in is the interactions between microbes and other microbes, microbes in the environment, and microbes and macrobes, for a better word, us, bigger organisms. And I'm particularly interested in that because life on our planet has been around about 3.5 billion years or so. For most of that history of life, it's been microbial. The microbes are the ones that invented life on this planet. All the genes, all the tricks, everything. Without them, we wouldn't exist. And so I'm really interested in how microbes that spent 3.4 billion years all by themselves doing just fine, and then the bigger things evolved and came along, what's the interactions between there? Because we really depend on the functioning of those microbes. So that's my interest, and that has been things like nutrient cycling and gas cycling and climate change. Most of the CO2 cycling that's happening on this planet is accomplished by microbes. How do we fit in? Those kind of connections. And one of those kind of connections is the interactions between disease-causing microbes and bigger organisms. So that's how I'm sort of interested in the question of disease. And I thought, right now, as we're sort of still in, but exiting, hopefully, the back end of a global pandemic, the COVID-19 pandemic, there's such a awareness now. We all have a really fresh awareness of what pandemics are and how devastating they can be, that I thought this would be a good time to talk about pandemics, not just COVID-19 and not just in people, but in the ocean as well. So let's talk a little bit about humans again and sort of our awareness about pandemics. For all of human history and well before we ever started to write things down and remember, there have been pandemics, okay? The first one that I could find records of that are well documented back in the Roman Empire was probably smallpox or measles, something like that. The descriptions weren't all that great, if you can imagine back then. It killed estimated about 5 million people. That's about 10% of the entire Roman Empire at that period in time. There have been, of course, you know about the Black Death in the Middle Ages that plunged human history into the Dark Ages for centuries, a couple of centuries. All the way up through the Spanish flu after World War I, HIV/AIDS epidemic, and most recently, COVID. And if you look at those numbers a little bit, COVID actually, 7 million out of the 8 billion people on our planet right now is actually a kind of a small number, okay? Gives you a little bit of perspective, at least it gives me pause to think about what it must have been like to live through the Black Plague or any of those earlier plagues that killed really significant parts of our population. So disease plays, and always has played, an important ecological role for all organisms, for all life on our planet. It's a balancing force. So if one population gets too high, it's ripe for a disease to sort of knock that down and make space for another one. So the importance of the disease, if we didn't have it, if we really wiped out all disease, we would be in a real problem. And in fact, human population is at that sort of point right now where there really are too many of us on this planet. And we have to really figure out, other ways. We have to put in other kind of management practices to manage our own population so that the planet can support us. Okay? I don't think it's a coincidence that there do seem to be more and more and more new, potentially pandemic-forming microbes coming into our population. The math of it is really quite complicated, but really, and I got this wrong, it's so simple there, but it should be a minus in there. Birth minus death, if it equals 0, you have a stable population. And that's really what all that the math up there is, really, and you can add reproduction rates, growth rates, metabolic rates, all of those things fit in there. But in the end, if you want to have, if there's to be a stable population, the amount of births that you get have to be balanced by the amount of death. And disease is an important component in this equation. To the point that microbes are mostly beneficial. Okay? Our life on this planet really could not exist without them. And that's true in our own bodies. Our own bodies, there's about 100 trillion microbes in your body right now, mostly good ones, doing things that if they weren't there and doing, you would be very, very sick, if not dead. Okay? And we're learning more and more about the role of the microbiome in the human microbiome and also the ocean microbiome, the Earth's microbiome. By far, the microbial components of our system are dominated by good microbes. And many diseases, actually, we're learning as we learn more about our own microbiome, many of the diseases that we suffer, some estimates are 90% or that kind of level of the diseases that we humans suffer from are not from our genes or not from bad microbes coming in, but are actually what happens when our own microbiome, something happens in there and they get out of sync and out of balance. Okay? You take a lot of antibiotics and you get a yeast infection. We do something to throw off the balance of our own microbiome and we get sick. Okay? And all sorts of diseases, cancer, heart disease, liver disease, kidney disease, diabetes, brain disorders can be linked to what's called dysbiosis, when our own microbiome gets out of balance. And one interesting point about that understanding, which is really just emerging now, past decade, maybe 15 years, we've started to understand how important our microbiome is that if you think about it, it's a lot easier to manipulate the microbiome than it is to do surgery or to treat a genetic disorder or a disease in human. It's more manipulative, it's more accessible. So I think in our own medicine and the way we take care of ourselves, this understanding that's coming about of the importance of our microbiome in the system is going to lead to our general better health in the human population. And there's a lot of other benefits that come from microbes. We wouldn't have wine or anything fermented, chocolate, coffee, all of those are thanks to microbes. Most of our medicines, if they don't come directly from the microbes, they come from plants that were trying to fight off microbes, that learned how to make chemicals and compounds to defend against the bad microbes. So a lot of our drugs originally came from, and we've studied and improved on it in some cases, came from a war between microbes and other larger organisms. So don't underestimate, don't think they're all bad, because they're really mostly good. Anybody have an idea what that is? What are you looking at? Anybody? What's that? Plankton? Close. Bacteria plankton. So this is, under a microscope, magnified 10,000 times, about. All of these little dots, some of these are viruses, most of these are bacteria. There's a few larger cells in there as well. So you're looking at a drop of seawater under a microscope, and I stain that seawater with a stain that stains DNA. So these are what I call, in the fluorescent stain, UFOs, unidentified fluorescent objects. But they have DNA and they have symmetry, so we know they have their life. They have the sort of hallmarks of life. And this is what you see in a drop of seawater. So it's not only us that has a microbiome. Everywhere has a microbiome, including the ocean. And just like in us, the microbiome plays a really important part in how the ocean functions. In fact, if you look here at the faculty of the Skidaway Institute of Oceanography, look at the biology fact, very few people study the big stuff. Really. Most of us are studying the microbes, okay? Because they're really the base of everything. And if we understand what's going on at the microbial level, especially in the autotrophic one, the plants of the ocean microbes, they use sunlight, the energy of sunlight, to fix inorganic CO2 and do all the nutrient cycling. And when you make measurements of how much of the energy and materials that get captured at the microbial level, 50 to sometimes 100% stays completely in the microbial realm. Doesn't get up to any of the bigger stuff. Okay? It just cycles back and forth here. That's how our planet operated for 3.5 billion years. And maybe less than 1% makes it up here. Okay? So again, mostly microbes are good. And you can see in this cartoon, these parasites and these viruses, these pathogens, they're also important parts of this system, keeping that cycle going. Birth minus death equals 0 means stable. Okay, let's go back. Let's get back into the bad ones, the ones that cause disease in humans. Okay? And because I think that we have invested our resources, our research resources, our learning resources, mostly into taking care of ourselves appropriately. So a lot of what I do in my research is I look to the biomedical, the human biomedical world, for ideas, for technologies, for tools, and adapt them to studying the microbes in the ocean, because there are really so many resources there and so much knowledge that's been developed there. So these are just some of what I've made this sort of list. These are all some viruses, major, there are many more, but these are ones that are sort of major pandemic, potentially forming viruses, okay? Influenzas, COVID-19 is up there, HIV, which is also a virus, smallpox, polio, Ebola. These are all pretty familiar, right? These are the ones that we surveil, that we try to understand how we can contain them if they go, because they have the potential to form pandemics. And the reason why we're so worried about these being pandemic forming, there are many, many, many more viruses, but not all viruses have the potential to form a pandemic, is because of the way they're transmitted. Okay? You'll see that most of these are either airborne, they can go airborne, and one infected organism can infect many, many more, waterborne, and increasingly the ones we're worrying about are vector-borne, insects in particular, mosquitoes, cockroaches, beetles. All those kind of organisms can carry pathogens and can affect widely. So Black Plague, for example, is one of those. Okay? And what's interesting in our current times is that because our climate is changing, the ranges of where those vectors go is also changing. Okay? And so we're going to be coming and experiencing getting into contact with things that we normally might not have. So one commonality in these viruses that can cause pandemics is how they're transmitted. Okay? And especially in these vector-borns, okay, but also sometimes the airborne ones, because weather patterns and stuff move them around. That's why we have the flu every year. It's a different strain. Are sensitive to climate because that is changing. Bacteria and parasites and fungi, they're also in the mix here too. So things like cholera, tuberculosis, typhoid, Lyme disease, all of those have the potential of forming pandemics. And then parasites like malaria, the plasmodia, there's a number of parasites that also, because they transmitted by vectors, insect vectors, can form pandemics. And so we study those and we surveil them. So a commonality of these potentially pandemic agents is that they are usually poor diagnostics. So if we don't know that they're there, if they're new, and we don't have good tools for that, it becomes a danger that we don't see it and it spreads too much before we can do any kind of mitigation of it. The other part is if we don't know what to do if you get sick, if we don't have any treatments. Right? Sound familiar for COVID? We didn't have a diagnostic. It took a long time, a seemingly long time. Actually, it was really quite short historically to get a diagnostic. So we have eyes on the problem, and we didn't have any treatments for it. So, and the treatments we have were pretty crude and didn't work very well. And then on the side, this is the arms war that you always hear about. So pandemic forming agents, usually we have poor diagnostics, treatments are limited, and they're highly infectious. And the arms war is that these organisms, particularly the viruses, mutate and change very quickly. And because the more we try to control them and prevent the spread, the more they try to figure out how to become more transmissible, how to become more virulent. And that's the arms war that's always going on. So think about, as we transition into the marine world, what kind of diagnostics we might have for that. What kind of treatments? Are we going to treat the entire ocean? We have limited abilities to manage marine pandemics. Okay, so here's the question. The answer should be obvious by this point. Are viruses, bacteria, and parasites restricted to humans? Absolutely not, right? They are everywhere. And so this is kind of old now. This comes from a paper from 1999, already a review paper. When we started, this field has not been around very long, so this was sort of at the beginning of it. And Drew Harvell, who's at Cornell, and a pretty big name in the field, put this review paper together with some co-authors, and she just tried to document and collate, figure out how many pandemics have there been in the marine environment, what kind of organisms are involved, what kind of agents are doing that work. And this is a list she came up with, and you can see it's a lot of different organisms. You know, sponges and seagrasses and oysters and fish and seals and, you know, the list goes on, okay? All have had documented mass mortality events. Maybe they're not pandemics. And all of them, you know, there are all sorts of different fungi, viruses, bacteria, et cetera. Okay, so there are a lot of them out there. Most of them, we have no clue what they are, let alone have any diagnostics or treatments available. And the other thing she pointed out in this paper is that many of them, these events that happened, were related to some environmental problem. Weather patterns, or human or pollution, those kind of things, triggered these kind of events. So they're environmentally sensitive. Ruth, did you?
These are all relatively recent dates. One assumes that you go back a billion years ago.
Right. I mean, as long as the history of life has been going, most likely. But nobody was looking.
It's a lot for a short time.
Right. Yeah, we don't go back to the Middle Ages or Roman Empire periods. Although there have been fisheries, and there are certainly documented cases where fisheries just crashed. And those are probably likely related to, both overfishing at some level, but also a disease that came in. Anybody ever been to Tarpon Springs, Florida? Ever been there? It's a Greek community that moved, that historically, and they're still doing it now, fished sponges. Well, this was the formation of Tarpon Springs was a whole town, I can't remember which island it was in Greece, that was just doing sponge fishing. And there was a bad disease that wiped out the sponges. Somebody figured out they had the same kind of sponges, the west coast of Florida, and they moved everything. Everybody moved. All the families, they built churches, everything, and they still speak Greek in the streets there. And then in the 70s, that same ciliate came along, wiped out those populations, okay? And they were in the same place. They didn't find somewhere else to move, so they started a tourism industry. But then the sponges came back in the 90s, mid-90s, the sponges, so they started fishing again. So now there's a 1970s kind of tourism industry there. They have museums with dioramas in it and all that, and they're back to fishing though. So, all right. So what I wanted to do, I hope you sort of get the idea that there are probably tons and tons and tons of these events. But I wanted to go through like 3 case studies. Marine mammals, sea otters, coral reefs, and more locally blue crabs and shrimp, which I've been studying and working on, as sort of examples of what marine pandemics look like, what the effects of them are there any solutions, what we should do about them, raise those kind of questions that we can then have a discussion about afterwards if there is time. All right, so we're going to start with bird flu in seals, sea lions, and cetaceans, dolphins, and whales. I love this picture, right? Very cute. All right, so marine mammals get sick. They're very closely related to us. So many of the diseases and pandemic-forming kind of agents that affect humans, largely are viruses, also affect marine mammals. Okay? And so, for example, influenzas, the flu, is relatively common among seals and otters. And when they're detected, these influenza strains are often the same ones that affect us. So when there's whatever the annual flu going around, guess what? These guys get it too. Same things. They get, like a pneumonia-like symptoms, and it's always around. And so we haven't looked at that much, but pretty much since it's been looking at, we find these, and they have these names that are sort of meaningless, H7N7, H4N5, those kind of things. These are all known mammalian influenza viruses, okay, that are moving back and forth between mammal populations. Deer get it too, wildlife gets it too, and marine mammals also suffer from that. An interesting estimation or observation is that about 3/4 of all the diseases that we get are thought to have arisen in other animals. They're called zoonotics, okay? Where we get these new emerging diseases from are coming from reservoirs in other animals. That's why we often survey them. Bats are a really good example. Bats are especially interesting because, first of all, they fly and they move around a lot. But because they're very strange mammals, as you can imagine, but because they beat their wings so much, they generate a lot of heat, okay? And so bats have managed to figure out how to have elevated temperatures and live with it. So to us, they have a fever all the time. 104, 105 degrees is a normal active bat temperature. Then they go to sleep in the winter and it goes down to 80 or something much lower. It doesn't take as much energy to keep it up. And so those high temperatures often keep these viruses and things that are typical in mammals, you know, that have temperatures, body temperatures more like our own, keep them at bay. They're still there. So they're really important. They become reservoirs for these zoonotic emerging diseases. So that's one of the reasons why we look at bats for that. And there are all sorts of reasons that we see increases in the occurrence of these sort of emergent disease in animals. And because we think that most of the things that we get infected with are coming to us, are coming from these animals, that's also a reason driving why there are more emerging infectious diseases for humans. And those things include adaptation, the arms race, immunologic dysfunction, which is often related to the microbiomes. There's more and more of us, so we're moving things around and making all sorts of changes, environmental degradation, climatic changes. All of those things are pushing the emergence of infectious diseases. So again, relatively new field, but relatively recent. This is one of the earliest really sort of big events in marine mammals that have been documented in 2011 in New England harbor seal populations and the gray seals as well. Maine to northern Massachusetts started to see these sort of mass die-offs of these seals washing up on the shore. Okay? So this is just sort of a case of what was happening. So this is September to December, that period in 2011. 162 seals in New England died, apparently of pneumonia. An influenza virus similar to the avian flu, what's been circulating in birds, okay, not in mammals, seems to have mutated enough to be able to infect a mammal, okay? And this was the H3N8 strain, and it was circulating in birds at the time. And so it's what's called a spillover event. It came from birds, adapted to mammals. It was typically concerning because the kind of mutations that was in this H3N8 mutant that was found in the seals had very typical kind of mutations that would allow it to infect mammals, probably including us. The receptors that it uses to get inside a cell, a mammalian cell, we have. So we became very worried that this is a strain now that can come. To my knowledge, this has never happened. There haven't been a case of H3N8 avian flu in humans. But then, this past year, okay, there was another big outbreak. And it's not that this hasn't been around every year. They've been being monitored. But in 2023, there was a new one. Another big outbreak happened. It just started in June. This time, it's also an influenza strain, but it was this H5N1. Does anybody know, does that ring a bell to anybody? This is the avian flu that was first detected in poultry in China. It had to end up killing something like 50,000 chickens at a couple of plants that started to get this. The problem with this one is that it does infect humans. Okay, there have been documented cases of H5N1 in humans that have resulted in human deaths. So this is a nightmare. This is a pandemic nightmare. It's transmitted by birds, which are everywhere. It's respiratory, right? So when you cough, it goes out, and it can infect humans. Okay, so it's highly concerning. Okay, and clearly it's quite fatal. In 1 month, by the end of July, so June, July, so a month, over 300 harbor seals had been died and confirmed to die from this. And for sure, that's just the tip of the iceberg, right? There's probably a lot more out there that we're not detecting. There's really a handful of people that are the Marine Mammal Health Survey group. They miss a lot. And again, once again, the waterfowl were suspected to be the vectors for this. And it's spreading. Okay, so H5N1 is now detected with a little bit later in the summer. It was detected on the west coast, Puget Sound. In Morrison Island, there were lots of birds, about 2,000 birds that had died from this. So that's probably how it's been transmitted. And it's not only in North America, in the US. Okay? There have been now that we know what it is and we have assays for it, we can diagnose it, in Europe, in Russia, in South America, this same H5N1 mutating just the way COVID does. So there's all these different varieties of it, but it's basically this group of viruses is now global. Okay, so this is a global pandemic in marine mammals and birds with the potential of spreading to humans. Okay. In Peru, there were over 3,000 sea lions that died from this past winter. And again, the people are pointing at the great black-backed gull as the primary transmitter, the migrating species. Okay, so emerging infectious diseases in marine mammals. What are the implications? Well, there are huge implications for human health to it. What are the solutions? Not really sure. Probably managing bird populations and managing the mammal populations themselves, making sure they don't get too dense so the spread doesn't get too aggressive. We're not going to get rid of it. It's there. Just like we're not going to get rid of COVID. So there's all sorts of wildlife management implications. There's also implications to use these species. Following them is a really good idea for us to make sure that they don't spill over into humans. Okay? So as population management strategies, wildlife management policies are being adapted and created, increasingly now they're taking into consideration concerns about emerging infectious diseases. And we are not putting enough resources in that even close. All right, so the next pandemic that's going on right now I want to talk about is coral reefs. So everybody probably has heard that the coral reefs on our planet are dying. Okay? They're bleaching, okay, and then dying. It's a global problem. This is basically the entire latitude, longitude, environmental permissive region for corals to live in. And basically everywhere that there are corals, we're seeing these mass die-offs. There's about a third of the Great Barrier Reef left. The Caribbean corals are in dire straits. We might not have corals in the Keys in a couple of decades. They might be just all gone, okay? Because this is going on. So what bleaching is, so everybody knows that corals are animals. They're related to jellyfish. They're cnidarians, okay? But they're benthic, but they produce planktonic larvae that travel in the water. And what bleaching is the loss of their symbiotic algae, okay, that they maintain. The color is coming from photosynthetic algae that live in association with the animal part of the colony, the coral colony. And when all of those leave, for whatever reason, okay, the coral, although it's still alive, okay, turns white. Okay, it's a calcium carbonate structure, and that's what you see. And all the algae that had the photosynthetic pigments leave, the coral turns white, it's bleach. Okay? And we know that that bleaching is triggered by environmental stressors. Okay, particularly temperature. So these big die-offs, like what's going on in the Great Barrier Reef right now, is because of marine heat waves. The water temperature gets up a little high for a day or two or weeks or periods, these sort of heat spells that we have, and the corals bleach. Okay? But again, it doesn't necessarily kill them. So why do they die? And the thought really is that much of the death that we see, the mortality that we see associated with these bleaching deaths, is because now the corals are really, really stressed. And they no longer can defend themselves against opportunistic pathogens. And so what they're dying from, the bleaching is being triggered by the environment, but they're dying from disease. I just think this is a fantastic picture from the American Samoa. This is a reef. This is all called Acropora palmata, staghorn coral. It's a very important reef building part of shallow reefs all over the world. There's different species of Acropora, but this is one of the ones that's been most impacted by bleaching events. That's in December 2014. A year later, in 2015, you can see it's totally bleached. Okay? The coral is still alive at this point, but it's totally bleached. But by August that year, it was all dead. This huge, huge tracts of this reef. Okay, and so the model that we have for understanding how this works is called the disease triad. And it's basically this Venn diagram here, is that you have the host, and when it gets stressed because of whatever reason, but in this case we're talking about temperature and heat waves, and there are opportunistic pathogens around, you get into this disease state. And that's where what we think is happening. We push this system, we push the environment, the host, and the opportunist pathogen, where these circles could have been apart before. They've been pushed together and creating this disease state. So that's the model that we're thinking about when we're trying to understand what's going on and is there anything that we can do about it. And this was a real breakthrough. This isn't a particularly terrible, terrible disease. What you're looking at here are Gorgonian soft coral sea fans, okay? And these lesions on here with the purple around it are a disease, okay? This caused by, it was figured out by this fungi, Aspergillus sandowi, okay? But the really important thing, and this is in the mid-2000s, that was maybe 2005, 6, 7, 8, in the Caribbean mostly. This is also Drew Harvell's work, one of the leaders again, as I mentioned. But what was really important about this particular disease is that they were able to culture the pathogen. So now they had an actual pathogen and a cause, and they really can put them together. It allowed them to study what the disease process was, how the bleaching happens, how the death occurs. Okay? And so we've learned a lot how the immune system of corals function. So we've learned a lot from this particular system, even though it's not necessarily the most terrible of the coral diseases. And on the bright side of this, another interesting thing happened. So they caught, her group and then many others that sort of helped and joined in and started seeing it all over the world really, was that after a few years of this going on and really wiping out huge tracts of these sea fans on coral reefs, they started to see it go away. And they even started to see corals that had these kind of damage and big dead parts, but the rest of the coral would be okay. And now when you take this same fungi that they isolated and you infect currently growing Gorgonian sea fans, corals, they don't get sick. So the corals have a pretty powerful ability to adapt to some of these kind of things. So they've learned. Okay, so this is, again, the arms wearing. So far, this fungi has not figured out a way to bypass whatever the coral invented to get it. So they can recover, and they will survive. The Gorgonians are not going to go extinct unless, of course, it keeps getting hotter and hotter and hotter. And there are many, many diseases, and it's really usually pretty difficult to figure out what's causing the disease because there's so many microbes associated with these corals. But these are just some of them. We talked about Aspergillus already over here, but there's white plague and white band and skeleton eroding disease and white pox and brown. There's lots of these diseases around. Again, probably a fraction of what's really there. Okay? And one thing to look at, I've sort of put what the types of parasite pathogen they are, talking about fungi, bacteria, protists like ciliates. Do you see viruses there? We haven't seen or documented many coral viruses. But I absolutely don't believe that there aren't coral viruses. I think it's more of what we don't know. I would expect that there are plenty of viruses around. They're harder to study. They move through really quickly. A lot of times these corals are dying. We don't even know they're gone. But the ones that we study are the ones that sort of have this slower progression, like the aspergillosis by Aspergillus. You sort of see it, and when the researcher just happened to have planned a trip to the Caribbean at that point, probably 3 years ago, they planned we're going to go there, and they come across that, it's slow enough for that to happen. Okay. So this is from some work from another group that took the time just in 2022, so this is a relatively recent paper, took all the literature that they could find about coral diseases, looked at it, and came up with what we think are the most important, the major diseases, the ones that we should probably be focusing on, thinking about what to do about. And you can see that about six different diseases out of all the ones that are out there account for about 3/4 of all the coral disease that we see on our planet, and 3 of them in particular, all bacteria, okay, are sort of the main ones. So that really helps us focus on the problem. And you can see again, these qualify, in my book at least, as a pandemic, especially the bacteria. If you look at the global distribution of these 3 top ones, these are the bacteria, they're across the planet, across the globe. Okay. Some of these ones are these ciliates, and bigger, more eukaryotic, more complicated animals are more localized at this point. They're just in the Pacific, but not in the Atlantic. Of course, if we start having an aquarium trade that brings corals from the Pacific to the Atlantic, and people put them in their aquarium and they have water flow through water in Florida, guess what? We're going to spread it around. But bacteria seem to really get everywhere. And this is also some data. I didn't have time. There is a huge program in Keys National Marine Sanctuary that's doing a lot of surveying. This program is continued. It started in the mid-90s and it continues to this day. But this is some early data when these sort of pandemics were just getting going. And what you're showing is, so 3 years of data from 40 sites across from the Keys up to just the Panhandle. I don't think there's Gulf of Mexico sites in here. The proportion of stations where they encountered disease went from like 20% or less, the black line here is the total, up to over 80%. Okay? And this is where I'm saying the corals are decimated. I suspect, however, even though we have a lot less coral reefs, healthy coral reefs in Florida and the Keys right now, I suspect this number actually might be down, okay, because we've wiped out a lot of corals, so there's not as many corals there, but what's left may be adapting and may be becoming resistant to some of these diseases. So, and I'm sure someone else smarter and more dedicated that's funded to do it is doing that kind of work, is plowing through and mining all this data that's being collected, mostly by NOAA groups. And I want to leave you, before I leave coral reefs, with a quote that's in Drew Harvell's most recent book called Ocean Outbreak, which if anybody is interested in this more, this is a really good read. It's a very easy read, and look for it. It's on Amazon for not much, and it's a nice book. And she sort of describes her adventures of all this work in a really kind of detective story kind of fun way. But she says the world's coral reefs, our most diverse and valuable marine ecosystems, are being sickened by a variety of factors all at once. Our only chance to limit their loss lies in understanding how all the threats to corals interact with and affect each other. So that's that disease triad. We need to understand what all those interactions are happening that results in this disease state, and what can we do to mitigate those problems. All right, let's finish up with some more local problems that have to do with the primary fishery species that we have in Georgia. Okay, and this is just the pie chart of the economic value of Georgia's fisheries, and you can see the two primary commercially valuable species are shrimp, primarily white shrimp, but a little bit of brown shrimp, and hard-shell crab, blue crabs. Okay, and we have other fisheries, we have oysters and all that, but they actually make up quite a small fraction of the value of our fishery. And guess what? Both these species have a lot of disease problems, and they're probably representative of pandemic kind of diseases. Although they haven't been studied that well, of course. So we'll talk about the blue crab first, and this disease called, colloquially known as bitter crab disease, also can be called hematidinium disease. Hematidinium is the organism, the parasite that causes this disease. Okay? And this is from a publication that Dr. Lee and myself published back in 2004. We were just coming out of a big drought, which we think is the environmental trigger of hematidinium disease, bitter crab disease here. And it's this parasite, which has a kind of a complicated, but it doesn't have other vectors. The primary host is the blue crab of this parasitic dinoflagellus, so it's related to the algae that make red tides. It's that kind of organism, and it can consume, it doesn't photosynthesize, even though it's an algae. It consumes organic matter. It's a parasite, and it can consume, in this case, the crab from the inside out. But it has this kind of complicated life cycle that includes, as a part of its life cycle, a free-living mobile form. So it's waterborne. Okay? That's why it can be a pandemic. Okay? And then it has this, this is the growth stage. This is where it feeds. So when you look at a sick crab, you see this trophont form, this feeding form mostly. Okay, so it's a protist parasite, it's waterborne, it's pretty indiscriminate against crustaceans. There are many species of crustaceans that are affected by this, and actually it might be that there are many, many species of this parasite, but it's very hard to study, and we don't really know that. We actually only know 2 different species, and then all the other reports of that say hematidinium species. We don't know what it is, but it's related to this. And it's global. Okay? We have it in our blue crab, as swimming crab, Callinictes sapidus. It's up in Alaska and in the Pacific, in the snow crab and the tanner crab. Did anybody notice last year snow crab prices tripled? Anybody notice that? Had a big outbreak, and you couldn't catch snow crabs in most of the Alaskan fishing grounds for about a year. And now they're sort of back. The cool thing about these kind of animals is they have incredible reproductive capacity. They're very fecund. Up in the North Atlantic, on the Norway side, the Norwegian lobster, which is actually a shrimp, also is affected by this, and mass die-offs of those guys. In Japan, the Japanese swimming crab, in Australia, related kind of crab. All of these guys get hematidinium. It's global. And this is what it looks like in our own fishery. So this is the DNR collects data, fisheries stock assessment data, and this is the blue crab data dating back to 1956 when they started. And this is pretty typical. So the yellow bar here, that's indicative of 1 standard deviation. So that's just the flux back and forth. And these are complicated to understand population data from fisheries dependent fishing, because it's how many boats are out there, and how much does fuel cost, and how much does insurance cost, and kids don't want to do this thing that their grandfather did anymore. All that factors into this up and down, but it's been relatively stable in there, except for 2 periods. There was this place in the late 60s where there were mass die-offs, and that turned out to be a fungus that eroded the shells, same kind of thing that often affects Maine lobster, gets the shell disease. And then when we had that drought, okay? And this was, I came to Skidaway right here. And all of a sudden, blue crabs, you could practically walk across the crab, buoys in Wassaw Sound. There weren't anymore. The crabs were just gone. And so, as a person who was interested in interactions between microbes and bigger things, DNR came to us and said, can you tell us what's going on? And so we started looking at it, and we found that it was this Hematodidium species, and we had bitter crab disease going on. And it wiped out. In Wassaw Sound, there were literally no blue crabs. You couldn't catch a blue crab. You put out traps, they wouldn't be there. We started to see other smaller crabs that usually would get terrorized by a blue crab because they're very belligerent. Blue crabs are. They started to come, the whole ecology started changing. They're a keystone species. The good news, I didn't have a chance to plot out and dig up and plot out the new data, but we're right back up here now. Okay? So they're very, if the problem goes away, they have the capacity to recover.
What made the problem go away?
What's that?
What made the problem go away?
Good question. The drought, we think. The drought went away. It started to rain again. Okay? And whatever it is, however that stresses them out or whatever that creates, we think that's the driver of Hematodidium disease. So we expect that the next time we have a drought, which we certainly will get, and we'll probably get them more frequently with climate, with our changing climate, this is not gone. It's just going to come back. Okay. But that's another thing. So, when it was a problem, we were getting resources to study it. But then the problem goes away, and so do the resources. So we're dark now. It's invisible to us. We're no longer able to survey for them, even though now we have the diagnostics for it. So what you're looking at here is, under a microscope again, stained tissues. These two are in the vascular spaces. They have a semi-open circulatory system, crabs and shrimp do. So we're looking in what's the blood is hemolymph. It's a combination of what our red blood cell and white blood cell is the same cell in crustacean. So it's called hemolymph, not blood. But all of these things, these purple things, all of those are the trophont form, the feeding form of the parasite. So these things are just consuming from the inside out all the crab. And this is the immune system. These nodules, hemocytes, are the crab defending itself. So the crab can defend itself, but it gets just overwhelmed. This thing is so prolific once it gets going inside a crab. It just overwhelms the immune system and the crab. This is fatal to the crab. In a day or so after infection, and you can transmit this infection either by having them eat an infected one or injecting it into their hemolymph, and then they die. So just like that, the coral, the sea fan disease, aspergillus, we can do that with Hematodidium. So it's infected. It's also waterborne. This is inside the heart of the crab. This is a piece of cardiac tissue. These are the reproductive forms. So these are the spores getting formed, and they're going to be released when the crab dies. And these are the cells that are producing all these.
So if people eat those diseased crabs, are they going to get sick?
Absolutely not. That's an excellent question, and I didn't put it up for the crab, but it's also going to be true for the shrimp. These things are very specific to crustaceans, and they don't affect humans. So unlike the viruses that we talked about in mammals, that because they're so close to the mammals, they're likely that we could get sick. That's not the case with these crustaceans. So you wouldn't want them to eat it, though. You would probably spit it out, which is where the name bitter crab disease comes from. It would be not very tasty. And I have to say, I actually have never eaten one that had it, so I don't know. But that's what I've been told. So we had, during that drought, we had a really interesting, I got 5 minutes, a very interesting natural laboratory to work in because of the way, so this is Wassaw Sound, we're right over here, Skidaway Island. Just south of us is Ossabaw Sound. The Ogeechee River feeds into that. So there's a lot more fresh water coming into Ossabaw Sound than there is in Wassaw Sound. So in the drought period, it got a lot saltier in Wassaw Sound than in Ossabaw Sound, and we saw a lot higher hematodinium. And that's how we started to put together the connection with the drought in this case. So we did see some hematodinium, but we didn't see as much as we saw it in Wassaw, and we did all sorts of experiments to back that up. The other thing that's really interesting is that blue crabs are incredibly adaptable. Okay, they can live in fresh water, they can live in full-strength ocean water. So the male crabs, mostly, and all the crabs really, but they disappeared from here. I said you couldn't catch a crab in Wassaw Sound, but there were crabs up here and up here. They went and they hid. And places where groundwater came, the little ponds that got formed that had groundwater, still had groundwater, full of crabs. Okay. But the females, in order to release their eggs, the eggs won't survive in fresh water. They have to be in full strength. So the females, once they get fertilized, have to make the trip out into the ocean to release their eggs. And so we really changed the sex ratio of the population, which has potentially long-term effects. That we expected to see that. We actually didn't see that. They just came back with a vengeance after it started raining again. All right, I'm going to hurry up a little bit and just talk about black gill disease in shrimp a little bit. If you guys have heard me talk before, you've probably heard me talk about black gill. I recently did a TED talk about it, so you can look up TEDx Savannah and you'll see me there talking about shrimp. This is another potentially pandemic for shrimp. All panade shrimp, the ones that we eat, can get infected by this parasite. That's what it looks like. Here's really quite, clear that it's there. One with, one without. Under a microscope, it was fairly. This is one of the first. Again, DNR came to us and said, "What the heck's going on? What's going on with the shrimp? What's causing this?" This is one of the first examinations that we did. This is a piece of shrimp gill tissue, thin slice under a microscope. These are all the hemocytes, the blood cells. At the tips of the gill tissue, these things are definitely not shrimp. Okay, it's a parasite. Turns out to be a ciliate. Nobody had ever found that ciliate. This is the immune response to it. The crabs mount an immune response, but if they become overwhelmed, again, just like hematodinium, then it could be fatal. And that's what it looks like. Again, it's probably been here forever, always been in our waters. Something triggered it, and now it turns into an epidemic. Okay? But no one had ever described it. This is a brand new species to the literature. And it's local, at least locally, it's an epidemic. So it's an epidemic proportions off of our coast still, and also now in the Gulf of Mexico. A lot of the lighter blue, it's present, but nobody studies it, so I can't say if it got up to sort of epidemic proportions. And we haven't looked everywhere where there are this group of shrimp, so we don't know. But I suspect very strongly that this is global as well. And for the fishery, this was kind of interesting data. So this is landings, and it affects largely the fall population, back from 1980s up through, this is current, fairly current. And you can see there's this big dip in these landings, okay? It happened somewhere around 2000, maybe a little before, right when we started to see black gill. So that's like a smoking gun, right? But causation, the correlation is not causation. So we've actually been spending a lot of time, and I'm unfortunately not going to have time to go into it, to talk about that. We are still struggling to show that this big decline in the fishery is actually due to this parasite. So we're struggling with that still, but there is a correlation with the winter water temperatures in the year preceding the fall harvest when we see the effect of this parasite. So there is a climate connection. Our winters in particular with what we're experiencing with climate change is warmer winters, and that is correlating pretty significantly for biology, based especially on landings, which is all, there's so many things that go into it. Winter water temperatures are pretty well correlated with the harvest that we're getting. So there's a smoking gun there. We haven't figured out the whole story on this. And I think I'm going to have to stop there, unfortunately. Let me just leave you with Captain McKenzie.
Well, I'm catching plenty of shrimp because there's nobody out there fishing on them but me. But it really don't mean nothing because the price of shrimp is down. And we know the fall shrimp's going to have black gill in it. And that black gill gets in them. It kills them before they get to us. And, you'll go out there one day and you'll be tearing the shrimp up. 2 days later, they'll be gone. But they just die out. They get weak.
All right, I'm going to leave you there tonight and open it for discussion. Mike Sullivan took that video a few years ago. So thank you, Mike. I use it all the time. I think he just says it just the way it is. So we really still don't know. There's a lot to learn, and that's, I think, bottom line about marine pandemics. We don't know. Thank you.
Do the photosynthetic algae with the coral, do they have more of a, say they get stressed when they leave, are they, do they contribute to kind of immune resistance or immune function in the coral, or do they help the coral in some other way that keeps them less stressed? Or what's that relationship, I guess?
Well, they do. They produce a lot of the energy and nutrients that the coral thrives on. So when they go away, the corals can eat, okay, but they generally, in many species, do it very little. They depend on their symbiotes, their photosynthetic, for their nutrition. Okay? There are probably, and this is not as well known, very important interactions with the coral microbiome that then affects the health of the microbiome. That the Cymbidium, that's the algae, the symbiotic algae, that Cymbidium helps mitigate. So all these corals have this very rich microbiome, and we know that when that microbiome goes into dysbiosis, it gets unbalanced, the corals get very sick and can die. So part of the death might be that the microbiome of the coral is messed up because their symbionts have vacated.
Talking about human illnesses. About 10, 15 years ago, there was West Nile virus up in the northeast New York metropolitan area. It was an issue for a couple of years, and then it seemed to have gone away. Is that related to anything, or is it just a natural sort of die-off of the virus?
A couple of things. It didn't just go away. There are still cases and cases of West Nile. It became sort of in the news, and we were all quite aware of it because it was new. Okay. We now have surveillance for it. We now have treatments for it. And so it's being managed. It doesn't mean it's not there. It's mosquito transmitted, and so all the, we're controlling mosquitoes, but mosquitoes are moving around. We've adapted to it, but it's still present and still there, and still could get out of control. It was on my list of, I don't know if you noticed that, it was one of the viruses on that list of potential pandemic agents. So it's still around. And that's what happens, for humans, right? Something new comes, it catches our attention, new and shiny. We have a lot of ideas and thoughts and methods and procedures for managing these things. We get on top of them and we get control of it, and we don't worry about it anymore. It's out of our conscious, old news, we forget about it, right? We move on to the next one. West Nile, and that's dangerous because these things are always around. And so we really we're very lucky that we have an infrastructure of really competent epidemiologists that are paying attention to this kind of stuff. It's why we invest money in being in Asia to do surveillance and those kind of things. And a lot of that is moving just because of atmospheric weather pattern comes to us. So it's still there. It's still a threat, and you could still get it.
Thanks.
You started off with talking about equal number of births and deaths is equilibrium. So when something like the black gill kills off all the shrimp, what does it do? Does it die them off?
That's a really excellent question. So we never saw, if the shrimp are really dying, the way like Captain Mackenzie says, they're catching them and you're tearing them up, and then next day they're just gone. Right? Where did they go? We don't see dead shrimp. So maybe they're getting eaten by predators or that kind. But we also don't necessarily see any big boost in their predators. Of course, everything eats shrimp, fish and crabs and whatever. So we don't really know what the ramifications of that. They are definitely a keystone species, but something like when we saw the crabs die off, what we saw was other smaller crabs, their population starting to increase. So
But I wonder, what happens to the microorganisms?
the microbe goes back down to sort of balanced levels and is now no longer at these huge concentrations probably. Is that what you were asking? Sorry. Yeah. But the microbes probably, they right, if their host goes down, then they go, that's part of the balance.
Kind of like the Black Plague that wipes up 250 million people and then
right.
transmits it.
And then it sort of faded out, right? Without having, they had no idea what that thing was.
They didn't have erythromycin back then.
Yeah.
But they just thinned the population, but the fleas still have it in certain areas of the world.
Right.
They're still combined.
And we probably reached herd immunity to it, and like the sea fans did to the Aspergillus at that time. But it took civilization a long time to recover from that one. All right. Well, thank you for listening. I know I went a little bit over, and I appreciate you coming out. And I hope I expanded your
Thank you very much,
thoughts.
everyone. Please be careful as you leave. We've got a couple of cars positioned